seminar afterwards. But this has been a real game changer in trying to understand the community of different HABs in the environment. But it's a pleasure to introduce Don. I'm really grateful for him coming and being here. And this is a big honour to be one of our distinguished guests as part of the Wiese program. So it's really nice to have him. So I hope you take advantage of his expertise and experience in the sciences. I think you'll gain a lot of information from him if you just ask. So Don has a varied background, which he'll talk about a bit more this afternoon in his workshop. But he actually has an engineering background and did his bachelor's in mechanical engineering, then a master's in civil and environmental engineering, and then his PhD in aquatic sciences, but it was still in the civil and environmental engineering department, all at MIT. Really prestigious school, very strong program. But all of this background and experience has really still exists in his research today, because he's doing a lot of technology development for forecasting and HAB monitoring and so on. So this background has been critical to that. Don has done a lot of work in the lab, so we see him, in my field at least, as the, we call him the Don, because he is like the godfather, we like to call him, which caught on a trip in Cuba, where everybody called him the Don. But he has done a lot of groundbreaking work himself in the lab, so this is not something, and he still likes to get in the lab. This is just taken last year, so he's still engaged and involved and likes to get his hands dirty. He's often in the field. He's been recently on a cruise in Greenland. So it's really hard to get him away from the active science, which is what's really great about him. The other thing I would mention is that Don is one of those people that is still excited about research. And I think the most exciting discussions we have is when Don will be working on his laptop and taking some notes, and then suddenly turns around with a big grin on his face and he's like, this is great, and the discussion just continues and everybody gets excited. So it's really good to have him on any team. He's had the opportunity to go lots of places, lots of icy expeditions to Antarctica, Greenland recently, but a couple of times I think to Greenland. Which is really great opportunities, but these are just highlighting two of many expeditions that Don has been a part of as part of his HAB research. Some of these involve tropical expeditions. So at the top here, in Cuba, enjoying a drink after a long day, the US Virgin Islands, Florida Keys, and the islands around Kiribati, which is sort of in the middle of the Pacific here, sort of south of Hawaii. But he's been to every place in between, so from the tropics all the way to the Arctic and Antarctic, and I think just about every continent in the world. Well, every continent in the world. So a lucky man. Lucky to do it, but also he's provided a lot of experience and expertise to people that really needed it. So a lot of these coastal environments, this looks like a tough gig, right? You get to go to these beautiful places, but actually these are places that have, endemic issues with HABs. So in the tropics, usually ciguatera fish poisoning is a problem, but in other areas, we have lots of harmful algal blooms of different kinds, which he'll mention. As part of all of this work that he's done, Don has received a lot of awards and accolades. I'm just going to highlight a couple of them. The Yasumoto Lifetime Achievement Award is actually the highest accolade in our field, and it's a real honour to receive this award, and Don was certainly deserving of that. That's from the International Society for the Study of Harmful Algae. Another was for all of his work that he's done throughout Asia, which have many problems with HABs, both cyanobacterial, so blue-green algae, as well as red tides and others. So this award was the President's International Fellowship from the Chinese Academy of Sciences. So again, an international award, but this recognition of the efforts and achievements that Don has been a part of. And this is a really important award. This happened just this year. Don received the 2017 Bus Wit Ketchum Award, and this was an award given at Woods Hole, but it's actually an international award, and it's really prestigious. So, really important achievement. It's really nice to see Don recognised for his efforts. And this award is really not just for the science and pioneering that he's done in HAB research on mechanism and modelling and such, but also for communicating that science. Which is really important, and important for all of us to recognise that communicating what we do to the public, to government and others is just as important. But he's very good at enjoying himself. He's an avid golfer. He's been all over the world. When he can take a break, he'll be golfing, and is really, really good at it. And as you'll see from the US HAB meeting, I mentioned this last night, he does like to let his hair down and have a good time. He's known to be on the dance floor every HAB meeting, which is really, really fun to see somebody that's so busy still able to enjoy themselves. And this is a shot here over Greenland, and just shows the beautiful places that you can go with research if you put your mind to it and work hard. You know, you get to enjoy just looking out off the ship. And I just want to mention before I go on, some of the work, you know, that allows this is the fact that Don has been involved in a lot of work with Congress. He's going to mention this this afternoon as well, I hope. But he's put a lot of effort into being an advocate for science and for HABs, and trying to communicate to Congress, government officials and so on, the importance of the work, the issues at hand. And this is done not to try and get funding for himself, but for the entire community. So, you know, through agencies such as NOAA, EPA, FDA, all of these things. He's been a big proponent of getting some of this research funding available to everyone. So my research has benefited from the heavy lifting that Don has done in Congress to make this an issue that stands out, that we see. But this last slide, which I mentioned last night, really reflects the Don I know and love. In his Gumby suit here, his dry suit out on his expedition in Greenland. And that's the grin I'm talking about. When Don's happy and excited about the work he's doing, that's what he looks like. So with that, I thank you very much for coming, and thank you.
All right, I feel like the old country singer here, right? With this is great. Thank you, Jeffrey, and thank you, Allison, for the introduction, and thanks to whoever the nominators were that helped me get this. Very much appreciated, and it's an award that I'm both humbled and so grateful. The list of Wiese scholars is quite impressive if you can ever read down the list, and it's very, very nice to be part of that group. Now, first thing I want to do, though, is a quick, can you please just raise your hand if you were up at South last night? Actually, let's do it the other way around. Who wasn't? Sorry, I don't want to put you on the spot. Okay, enough were that I'm going to, last night I gave a big, long introduction about HABs in general and covered a more general view of the field, and then at the end got into a little more about what we do, but today's going to be a lot more nitty-gritty about my own research, but I do want to start off with some of the same introductory material, but much, much reduced. But if everybody had been there, I would skip through the first few slides, but I won't do that. The first thing that I wanted to say is that the title of my talk is talking about new technologies in HAB, or harmful algal bloom research, but another, if this would work. Oh, you know what? This were you using this? Is this working? It's not. Let's just try that. No, the laser works. It's the advancing. We're not advancing a slide. I tried the keys. They're not working either. So maybe we restart it here. We're frozen. It's not. See, I wanted to use my computer, which of course is a Macintosh, but they wouldn't let me. I have to use a PC. So, but anyway, I can at least tell you what the next slide was. It was a similar. There we go. Does it work now? Okay. So this is the title that I gave Jeffrey some time ago, but in truth, I think this is a much better title for what I want to talk about, which is novel insights into HAB dynamics using in situ instrumentation. So I covered last night a lot of different kinds of technologies, but now I'm going to focus exclusively on a couple of the ones that we use that are in situ sensors. And it's an area where I think not just HAB science, but oceanography in general is going, and it's an important area to highlight. So again, by way of background, the topic is harmful algal blooms. Down here you typically might call them red tides. This, in fact, is a picture from Texas of one of the organisms that can affect you here. And it, let me see here. All right, this is not the one we had last night. So what you see in this particular image is the water is discolored by the accumulation of cells that grow in enough density to change the color of the water, and in this case they are producing toxins that are killing these fish. And if you go along, I'm not sure whether it happens here, certainly along the west shelf of Florida, then you can find millions and millions of dead fish. These are caused by what you call your red tides. You also have these other symptoms, aerosol. The toxins can be aerosolized in sea spray and cause a very difficult time breathing along the shore. I could go on and on. There are all of these different types of HABs. The ones, we're going to talk a lot about this one here, paralytic shellfish poisoning. Notice these first 5 are all related to shellfish that filter the algae from the water, the toxic algae from the water as their food, accumulate toxins, and then are dangerous to human consumers. So even though they're called, shellfish poisoning, it's not the shellfish that are being poisoned. They're doing just fine. They're almost unaffected by these toxins, but it's humans and others who eat these shellfish that get the poisoning syndromes. And you can see that they're really varied. Some of them are lethal, some of them are not. This one's always interesting for people to hear, the amnesic shellfish poisoning, or ASP. You have a hint of it down here. The organisms that cause it are around, don't seem to be too terribly toxic. I do hear that you are finding domoic acid in manatees, and it's here. That's the toxin that causes that. But it's of note that particular toxin is one that it can kill you, but the worst thing, in a sense, if you survive, it can cause brain damage. True brain damage that is permanent in the short term, the area where you store your short-term memory. And there's many other sort of versions of HABs. Ciguatera is something that I work on with Allison in the tropics, tropical reef fish that become poisonous. Again, the source being single-celled algae that are the base of the transfer of their toxins up the food chain as they are eaten by herbivorous fish and then by carnivorous fish eating those. There are brown tides. There's fish mortalities of many different types. I mentioned a little bit about desalination. I showed a lot of pictures about this incredible green tides of seaweed blooms that are just breathtaking in the disaster that these huge outbreaks of millions and millions of tons of seaweed washing up on the beaches in China in that case. So there's many different types of HABs. And in terms of our own research, one of the problems that people like me face is exemplified in this picture, which is of a community from the water of phytoplankton and other organisms. And the reason I show it, and I'm showing it again, is that in this picture, this is the organism called Alexandrium catenella, that I'm interested in. It's photosynthetic. You can see these little gold balls, but all of these other co-occurring organisms are the noise that are there. That's the problem in, if you're a phytoplankton ecologist, you're measuring productivity, you don't care. You measure how much carbon is fixed by this whole community. If you're someone like Jeffrey, you're worried about which ones are taking up the silicate. He's worried about a class of organisms. He's got some of the same problems in that he doesn't like to have my dinos there, but he's still able to look at a group of organisms. What we're trying to do is look at a single organism in this assemblage, and that is hard to do. And I'll try to explain how we're doing it, and in two different systems. We're sort of now back east, and of course Woods Hole is here, and this is called the Gulf of Maine. And here you see an image. It's a snapshot from a computer model, but it gives you a feeling of the scale of these phenomena, which can stretch for hundreds of kilometers along the coast and quite a ways offshore. Big, widespread coastal phenomena. But we also have over here a much smaller version of these kind of Alexandrian blooms. So if you take this little bit of Cape Cod here, where the pointer is, blow it up, this is what you see. It's what's called the Nossa Estuary, not but an hour from Woods Hole. And here you see the estuary, and then there are these little salt ponds, we call them, kettle holes that the glaciers dug out that are deep, relatively, maybe 10 meters deep compared to this very shallow system. And what this picture shows, and what you'll see more of, is that this is where these Alexandrium blooms occur in these systems, in these little localized spots. And that's where I actually did some of my thesis work, because they're just perfect places to try to study blooms. I mean, this is totally isolated. Everything happens there in one place. You can use rowboats, small boats, very simple. Nothing compared to the expenses and the difficulties of trying to sample something here where the water's moving, everything's going past you. It's a whole different ballgame. But there's reasons, of course, because on the left that's causing huge, widespread impacts. But what we're doing is using the studies here to inform us, to help us protect the shellfish and the population along that coast. So, oops, there's a black button here, isn't there? So, yes. So, one of the things that we have to emphasize then is the life history of this organism. And this is our Alexandrium catenella. These are two cells that swim through the water. They're the ones typically eaten by the shellfish that make everything poisonous. These are cysts. These are resting stages, dormant cells produced by this organism. And this life history sort of shows you, let's start with a cyst that's in the sediment. It germinates to produce a cell that goes through meiosis to produce these two daughter cells that then divide and give you the population that is the one causing the problems. But then at some point, and I'm going to talk quite a bit about this transition, these gametes are formed from these cells. They're smaller, they fuse together to produce something called a zygote, a planozygote, swimming zygote, which then insists and goes back to sediment. And this is one of those excitements of when I was a graduate student and I was doing what was water chemistry. We were looking at these blooms thinking there had to be something going on in the water that was allowing these cells to grow and become so abundant. And in the process of manipulating those cultures, I kept producing cells that looked like these. And that's when I went down to Woods Hole and talked to some geologists there who were studying things like this in the sediment. Then we went out and figured out a way to look in modern sediments and found these. In fact, this picture goes back that far. It's one of the first times anyone has ever seen the cyst of Alexandrium, and all of a sudden my whole life changed. The whole direction of my thesis changed because the biology of this is so fascinating. But where these cysts are found determines where the blooms originate. Their abundance can determine, in some cases, how large the blooms are. You can disperse this species. There's genetic recombination involved in producing these. There's many, many different aspects of the ecology of this organism that all suddenly were available for study once we made that discovery. Now, the reason that I showed some of this yesterday, but I can show you a little more detail. What many of us want to do is to try to forecast and understand these phenomena, these bloom phenomena. And we do that with a numerical model. It's a complicated one in the sense that it's a coupled physical, biological model. And what we first have is physics, and I guess I'm a little surprised there should have been, whoops. This has got too many buttons. That's strange. Well, let's just skip ahead. And this is where I'll just skip through. Yeah, I'm missing, there's a slide that's gone. I'm not sure why, but all right. So what I'm missing is a slide that was going to show you the hydrodynamic model that we have that is well characterized, the oceanography of our region. But here we have to add to it this biological submodel, and it starts with a cyst map that we produce. We go out, it takes a week or more on a big ship to go out and map this out every year. And this allows us to know, this is the inoculum. How many cells will there be there to start this big coastal bloom each year? It's like how many seeds of crabgrass or something you might have in your yard that might might grow during a year. We can count them. And then we have to figure out when they germinate, and that's what's shown here. It's actually an internal clock that these cells have that says that during this time of the year they can germinate. These are 4 years here. So this time of the year they germinate. This time of the year they absolutely don't. And in between there's this percentage that does. This is all parameterized in that model. So this says how many are there, when can they germinate. Here it's how fast do they germinate as a function of temperature, and then light versus the dark. And then once they've germinated and you have them in the water, there's all sorts of measurements we've made of growth as a function of temperature and salinity and so forth. And the result then is this type of a simulation. This is of a big outbreak in 2005, where you see the cells that have germinated here and germinated here from those big cyst seed beds growing, proliferating, moving down with the coastal currents that go down here. And so that's a very useful model. And one of the things that we're trying to do is to improve the use of that model, and especially its usefulness in forecasts, and that's where this instrument called the Environmental Sample Processor is being used. It's our effort to try to obtain data from fixed stations along the coast that is real-time data about that bloom that we can use to update that model and make it better. And as you see on the upper right, this is just almost like a dot plot, but it's a chemiluminescent array basically based on DNA of the organisms. And we are targeting ribosomal RNA with small little probes that allow us to say these spots are only being caused by Alexandrium, with the intensity of that spot determining how many cells are there. We could have up to 10 or more species being detected at the same time, and I'll show you how this works. And there's also an antibody procedure built into the front end of this module, where you can use antibodies to the toxins to measure not only well, three different Hab toxins, but then we get species and toxins all at the same time. So here's a little movie that shows you, we're going to take the ESP with its batteries and open it up. So here you see a syringe. First of all, we put a filter here. This is all done robotically. We pull in seawater through there, through that filter, capture these organisms that were there. Another syringe pumps in a lysis reagent that rips everything apart, liberates RNA and DNA. Then we have another puck that we call it with the DNA of our target organism spotted on there. And then we do a whole hybridization. It's all done robotically, underwater, autonomously, complete hybridization. As I said, a chemiluminescent detection. You pick up this puck, put it here, have a camera that takes a picture, and then you would have many different organisms on there that you can quantify from that image. And that, of course, is then radioed to shore. Very cool instrument. It's actually commercially available. You can buy them now. Now, in order to use it, we've had to develop a very strong system with handle the waves and currents and this bungee cord. I didn't mention this last night. I did say that this can stretch 2.5 times its length without being damaged, still having information go up and down there. We just deployed it this last year in the northern part of the Gulf of Maine, in the Bay of Fundy. You've all heard about the tides and the currents in the Bay of Fundy. Well, they destroyed 2 of our cables. They tried to stretch it 3 times its length, and we lost 2 cables this year trying to push the bar. I always try to take these instruments in places that are harder and harder, and 2 of our 4 instruments this year, the instruments collected data, but we couldn't communicate with them because the cables stretched too far. So we have to redesign them and find better ways now to deal with even harsher environments than we've used so far. But we're still doing well, I think, getting them out there. So again, this is all I've showed this before, but I'm showing it I want to emphasize something else this time from last night. We put these ESPs in several different years in locations right near where the state in red here monitors the flesh of shellfish for toxicity. And we get this kind of data where we have in blue then the ESP data. You can see it's very patchy. This is daily. The patches of cells are going by these instruments, in this case this one instrument. Here is the weekly sampling by the state of the shellfish toxicity. So it's low, it goes up, and then it comes down. The ESP is seeing low counts, higher counts, and then lower counts, so there's a lot of agreement there. And as I said, what we're trying to then do is to model this. I skipped through the details here, but it's really a simple one-compartment model that says we've got the toxin at any time point. We're trying to take the ESP cell counts, turn them into measurements of toxicity. Toxin at one time point is the toxin at the previous time point plus some constant times the number of cells we counted. That's uptake, minus some constant times how much toxin you had at the previous time point. That's your loss factor. And we go through all of these fits, curve fits, and so forth, and get this kind of data where you see a very great, in fact, extraordinary correlation between now ESP estimates of toxicity and these measurements of toxicity done weekly. So, but the reason I'm trying to show this again is to emphasize what's going on here and going on here. That the state says, oh, this is the quarantine level. This was totally safe shellfish. No problem to eat this, but that's because they sampled weekly. The ESP is saying that if they had gone out and taken daily samples, they very likely would have seen levels above the quarantine level. Notice up here too. Now, somewhere around 200 micrograms is when you start to feel the symptoms of the saxitoxin that's in here. That's the lethal dose is higher. It's closer to a milligram, 1,000 micrograms, and so it's not that people would have died here, but you're to the place where people would have started having symptoms, and who knows how high this could have gotten. And so the point that I'm making here. And it's the same story in this other ESP a little further up the coast, that again, there are times when we are seeing numbers that are higher than what the state has measured in their weekly samples. So the in situ monitoring in this case, and our estimates of the toxicity, reveals considerable patchiness in the toxin dynamics, including several intervals where weekly flesh testing failed to reveal potentially dangerous pulses of toxicity. So I'll give you an advance. Part of what my message is today is a lot of the things that we learn by putting instruments in the water that give us daily, real-time data is a whole lot better information than what we're getting with our old methods. And this is the first example with the ESP. And what I want to then do is go on to another instrument that I only mentioned by name, didn't show any detail, and this is a really cool instrument. This one's called the Imaging Flow Cytobot. I love the name too. It is basically a flow cytometer with imaging capabilities that can be submerged in the water, and it's an incredibly powerful instrument, and somebody's got to get one down here because it really is a good instrument and cheaper than the ESP, and in many ways much more diverse. Here's a little movie of how this one works. You can see this one's actually quite small. I can lift it up very easily. Here you see a syringe that's pulling in water from the outside. It directs all the cells and the water down through this flow channel, and as it goes through, the cells are actually in single file. There is a laser then that will be firing at the particles as they go by here that you will see. And all we really care about in this application are the cells with chlorophyll. So you see there's a cell, there's a piece of dirt, a sand or whatever. Sometimes there's just nothing, sometimes there's chlorophyll. Whenever there's chlorophyll fluorescence from a particle, it's picked up, it's triggers a camera, which then takes high-resolution pictures of that particle or that cell. And these are 8 or 10 megabytes per picture. They're high resolution. They're really good. That information can then be transferred to shore. There is not yet sort of onboard processing of it, and you can get these, and I'll show you much more of these incredible communities of organisms are out there. But you need what's called this image classification. Let's see if I can, well, I'll stop this now. But there's many, many more images than we can handle as human beings. 8 to 10 images a second. This is 600,000 images in a day, millions in the course of a bloom, much more than, most of us would say a joke and say, well, we'd just get graduate students to do it, but even graduate students couldn't do this in an economic way. You've got to use computers and image classification. You need the kind of software that is used in facial recognition in airports and so forth. And so that's what's been built into this system. And so with this is a dashboard of one of the output from one of these IFCBs during a bloom, and it shows the bigger cells here, tintinids and so forth. Here are a number of different diatoms. This one actually happens during an Alexandrian bloom, so these are these larger cells that we're calling planozygotes. These little ones are these gametes that I was talking about. But I'll show you other dashboards of other samples, totally different assemblage, but you want the computer to go through and take each one of these, and they've developed for 150 or so now different organisms, classifiers that allow the computer to look at different orientations and say, that's this particular species. And we have done that as well for the Alexandrium. You can see one of the problems we face in doing this, that this just shows 1, 2, 3, 4, 5 different stages, but there's many more. If you really look at the various cell types that we have to deal with and to try to identify using these classifiers in a field sample, it's mind-boggling. And I have to admit that when one of my students or then a postdoc said he wants to use the IFCB on these blooms, I thought, oh, I just think this isn't going to work. because of several things here. First of all, our cells are round and brown, and there's a lot of round brown cells out there, and we just didn't think that there would be enough resolution to identify the Alexandrium. Well, that's turned out to not be the case. We can actually do that. And not only that, you can see there's all these different types of singlets, little gametes, vegetative cells. These are these big planozygotes, the swimming zygotes. Then you've got doublets where the cells are paired. You've got triplets. You've got quadruplets. You've got dividing cells that are different from cells that are fusing. You've got cells that have parasites in them. See this white? That's a parasite that has infected these Alexandrium cells. These are cells that are breaking open, in some cases releasing parasites, but they're lysing. These are cells that have been grazed that are inside ciliates and other micrograzers. So we have developed classifiers, ways to tell the computer that if you see this, that's Alexandrium. That's 4 cells. It's not some other species. If you see this is an infected cell. And so you can imagine, well, I'll get to it. I added these because down here, you're not interested in Alexandrium. You're interested, and I can see Jeffrey's all happy because we got diatoms here. And this case, this is underway sampling. We've got the IFCB on a boat that's traveling at 10 knots, taking samples every 20 minutes. It takes 5 milliliters, and then you get images like this that you can count and analyze. And in this case, we've just put all the diatoms together. And you can see in this particular cruise, we were really interested in these pseudonychia cells, which are the ones that produce domoic acid. But you can see all of these others that are also being imaged at the same time. And another organism or 2 organisms that you would be interested in around here are easily recognized with this instrument as well. One of them up top is Dinophysis, produces the DSP toxins, and you can see this is now from Lisa Campbell off in Texas at Port Aransas. They put an IFCB on this pier, so it's not in the water, it's actually drawing water up to it. And here's a sequence showing Dinophysis bloom. Here's Karenia, the organism that causes a lot of problems here in the Gulf of Mexico and causes NSP as well as a lot of mortalities of fish. And here's some NSP or Karenia observances that in fact were not even observed by the state monitoring programs. They were fairly quick and although reasonably large, but weren't picked up by the normal monitoring program. But the real interesting part of this is that there's now been 7 HAB events that this instrument has provided early warning for in that region. And they now have 3 of them. 3 of these IFCBs moored in different places along the Texas coast, and they're networked, and they're basically watching for these HAB species. And when you get above a certain threshold, they send out automated email and text messages to the scientists, to the managers, saying something's out there, you better pay attention. And that's really, to me, one good part of the future of the use of these sensors is in early warning and detection. Again, Jeffrey said I should get a commission from McLean, the company that makes these, because I'm so keen on them. Look at what you could do. You can pick up cell cycle stages. In other words, if you wanted to study the division of diatoms or even tintinids, you can see that. You can see it happening here. You can look at sexual cycle stages, like the fusing cells of Alexandrium or oxospores. All of these kind of processes are visible in these images. You can see symbioses, parasitism. You can see predatory feeding mechanisms. This is a great image. I had a student do a thesis all about these Protoparidinium dinoflagellates that feed on a lot of diatoms using this thing we called a pallium, which is like a veil that it throws out and envelops these diatoms and then digests what's inside them. And it's this very diaphanous kind of a thing, and you'd think it's so fragile, but when it covers these diatoms, here it goes through the IFCB perfectly fine. You image it and actually catch this cell in the act of feeding. So what I'm pointing out is here I'm telling you about HABs and the focus we have on these, but there are many, many other biological phenomena in the plankton that you could study with this kind of device. All right, so I told you about those salt ponds or kettle holes. This is one of them, and in fact, it's called Salt Pond. This, if you ever go to Cape Cod, is the National Seashore Visitors Center that overlooks this area, and you can see why it's such a good place to study. It's large enough to have blooms. It's marine. It's connected to the ocean out here through this marsh, but it's connected through this very shallow and narrow channel, and that restricts the flow of cells in and water in and out of here, and allows, if there's cysts in here, which there are, the cells to bloom and then drop new cysts with relatively few cells being lost because of that channel. And I'll talk a little bit more about that later. And as I said, we developed 15 different classifiers for all of these different Alexandrium life history stages, and then we put one of these IFCBs out here in this salt pond. And what I'm about to show you is some of the findings that I made some years ago, which we've confirmed now, is that one of the reasons the cells go through their whole cycle in here, and until now we thought very few were lost to the outside water, is because they migrate every day. They swim up and down the water, but they don't go up to the surface. They go up to about 1.5 or 2 meters, which is in fact deeper than this inlet sill here. So the mechanism that's a retention mechanism for these kettle holes is that, during the daytime, the Alexandrium cells have come up to them, not really the surface of the water. If they did, they'd be lost through this inlet. Instead, they stay down below and they accumulate. At nighttime, they go down even deeper, so there's no chance really of being lost. So they're going up and down like this, but it's a restricted migration. And that's one of the reasons these populations are staying there so much. Now, here's another model. This is probably the most busy slide I have ever seen. I will try to walk you through it because it's a lot of cool information here. This is a model of Alexandrium and the hydrodynamics of that Nossa system. So here's the Nossa Marsh. I've mentioned Salt Pond. That's up here. That's what we're seeing over there. These are other areas that Alexandrium occurs. Remember I said in these little other distal parts of the system, this is where the blooms develop. Here's the open ocean, and you see water sort of flooding in and out of the marsh system, and some of it, gets into here and down into here. Now we're looking down on the salt pond here. This is, you can see that this is the days we're going up and down, the tides going in and out. Here's Alexandrium in the color is how many cells we've got of Alexandrium. Here's the temperature in the system as the tide goes up and down. And these cells, we gave them the behavior that I said, where they don't really swim up to the surface if they can help it. They sometimes get washed up here, and some of them do leave, but by and large you can see this concentration mechanism in action, where they are largely here, and it takes unusual situation for them to come out, but many of the time they come right back in again. So you can see that through time. Now there's a mortality function in here, which is one of the reasons this bloom is diminishing now. But you can see that in this area where we're developing the blooms, that we really do get a great retention capability. So this model, again, like our other models, is very useful for understanding the phenomena that go on there. So what has the IFCB told us? Well, first of all, we had to make our system quite diverse and capable. We have kind of an ugly raft. In fact, it's interesting, this happens. This is, remember, right below the Cape Cod National Seashore Visitor Center. They've been great having this research going on. We connect, we have a display about the real-time data, but then they came up to us this year and said, this is can you guys turn it into a boat or something that looks like it's just a dinghy that's moored out in the pond? So we now have to take what's a very functional platform and spend some money and fix it up. But what you see here is solar power. There is a generator here. In the first iteration of this, we had only a generator, and every 2 or 3 days we would send the graduate students out to put gas in it and sort of make sure everything's okay. And then now we put in solar panels, so we almost never have to use a generator. There's enough sun there to power this. You can see these winches down here, we have, well, I'll just work from the top. We have these solar panels for the backup generator. This is a Wi-Fi enabled platform, so you can see just there, I missed it, the CTD that we've got in the fluorometer comes up to the surface, instantly sends its information to the Wi-Fi hotspot that's on the raft, which then gets communicated to us. Then it goes back down. We are profiling using this winch. We can control it from our lab. We can say, you know, go down and stop, or go up and down slowly. It's profiling. There's two of them. We're not only profiling the CTD and a fluorometer, we're profiling the IFCB. And here's a great advantage of this kind of thing, because I just said Alexandria migrates. It's going up and down. If I just have a fixed instrument, then in fact the first deployments we had, the cells would sort of come up. All of a sudden we'd see a lot of them, and then we'd see more, and then they'd disappear. And your daily records were very cyclical. Now we can actually program that so that it moves up and down with the cells. So we're always there with the bulk of that population, and you'll see some examples of that in a second. But this is when you start seeing the incredible data that we are getting from this instrument. I'll try to explain here. Up top, imagine this just says cells per liter or something. It's a volume measure. It's a biovolume measure. This gray line are all of the other particles and cells that we're seeing there. The black is actually Alexandrium. Alexandrium does dominate this system during this is time here. You can see April, weeks in April here, starting in March. This line here is now showing the volume distribution of the singlets. Remember I showed you that one category that includes the vegetative cells before they divide. It includes the planozygotes, the big ones, and the gametes. So if you just count those, what you're seeing are several things here. First is that during this early, and the bloom is sort of building up here, you can see quite a level size distribution. This gray shadow, remember we're taking 3 samples an hour, 24 hours a day. That's what this gray shadow is. What we're showing you here is the day and the nighttime means of that. This is and first of all, what you see is that the cells actually get a little bigger at night and get smaller after they've divided, bigger and smaller. And what I'm trying to point out in this one, though, is, I'll come back to that, is notice how the cell size dramatically drops right here where the bloom is peaking. That these shadows down here, we've got a huge number of cells that are really small. And that is followed very soon thereafter by much larger cells. And notice that this sort of day-night cycling, which was division, isn't happening anymore. These are gametes that have fused, and they've made zygotes that aren't dividing. They're big cells, but they're not dividing. They're just getting bigger. So what you can do then is figure out the percentage of cells that made gametes. Now, if you had asked me before we put this in the water, what would we see, I would have said, oh, I bet we get 10 or 15% gamete formation. That's what we do in cultures. That's about how good we are. The number, though, is much higher. It's 90% or more of these cells are fusing. As much as 90% of the population is fusing there. So the observation is that sexual induction of Alexandrium catenale is far more robust in situ than in the cultures. Let's go back to that. I hinted at what was happening here. Cells get bigger, they divide, bigger, divide, bigger, divide. That's called phased division. And when that happens, you can actually calculate an in situ growth rate. Now, any phytoplankton ecologist in the room would know that that's been a holy grail in our field for the longest time. Jeffrey's nodding. It's like, it sounds simple. How fast are the cells growing in the water out there right now? Well, you tell me how to measure it. It's really, really hard. This is what we're doing here, because we've got phase division, we've isolated the cells optically and with the numerically to the ones that we know are dividing, and we can get an in situ growth rate. And when you get that, in fact, the numbers are really high. This is growth rate here. This is the temperature of the water, and remember, we know exactly the temperature where these cells are because we're going up and down with them. We're living with them with that instrument. And What this shows is during that bloom, the water went from, let's say, 5 or 6 degrees to 10 or 11 degrees. These are those in situ growth rates that we were calculating. These are all cultures that we have from that same area that we have grown in the lab and run through what we have as a temperature gradient bar, where we can measure the temperature, measure the growth rate at all these different temperatures. And you can see that the cells grow really well. Out here at 20, down here between 5 and 10, we're not growing very well at all. We're growing 2 to 3 times faster in the field at those temperatures. And you could say, oh, there's something wrong. There's something wrong with the way you guys are doing your math. Well, salt pond is very much like a flask. We determine growth rate in flasks by taking counts every few days, measuring the accumulation rate. Well, we can do the same thing in salt pond, and that's what this line here is. It's called the accumulation rate, basically, of cells in the pond. Now, that is a net accumulation. Some cells are being lost out through the inlet, some are getting eaten, some are dying, and so you would expect that it would be lower than the measured in situ growth rate of the dividing cells, but notice that it's much closer to that than it is to these laboratory measurements. So I want to emphasize then, cells are growing at least 2 to 3 times faster in the field than we thought they would be growing at those temperatures. Let's take a look at their swimming. Again, this case now, we're looking at the fluorometer that we've deployed there, and you can see how this daily migration happens. Here's the surface. Up here you can see the tides coming in. The cells are deep, not surface, deep, surface, and so forth. And if we just sort of look at this part of the bloom, you can actually calculate the rate at which that center of mass is going up and down. So we actually get a swimming rate out of it. So that layer is moving up and down basically at 15 to 20 meters per day. Previous estimates that we've had from laboratory cultures, from tanks and so forth, have been 5 to 10 meters per day. Someone asked me last night, these cysts that are down there at 70, 100, 110 meters, doesn't it take them a long time when they germinate to get to the surface? Well, at 5 or 10 meters per day, the answer is absolutely. That it takes a week or longer, and many of them are going to die in the dark because they're going to run out of energy. They're not going to make it to the surface. If, in fact, they can swim at 20 meters per day, then it's only 3 or 4 days we can have a lot better survival. So our understanding of this swimming rate is hugely important ecologically. But there's more in this image than just that. I should go backwards. This is when the bloom is sort of just growing and growing and growing. This is the end of the bloom. This is the time when those planozygotes appeared, those big dark cells, right? The ones that are the result of fusing gametes. And look what's happening. They are going up closer to the surface. This is when your advisor, me, wrote a paper years ago saying because of this migration and they stay away from the surface, the cells never leave the pond. This is why the ponds accumulate. And then the graduate student comes along and says, hey, you're wrong. You know, look at these guys are swimming up to the surface. And the answer of what's happening is that it's the life cycle stages that are different in the way they are migrating. So basically, the shoaling or the shallowing of this planozygote migration gets them up close to the surface. They get up close to the surface, what happens to them? They're going to get exported from the pond. That's the theory. So the cells swim progressively shallow in the water column over the course of that sexual period. So how do we prove, though? There's clear dispersal, species dispersal implications to that. Well, this is where, again, I mentioned this yesterday, we can take the IFCB and put it in something called a jet yak, which is a kayak with a little jet ski engine in it, and you can operate it with a little control box. It's totally remote. And here you see salt pond. And what we're saying is that cells are being exported out here, but this is really shallow. You can't get a boat out there very well, especially when you're looking at the ebbing tide. So we put a jet yak in here and started to measure what was going on. So these are just tracks showing, first of all, we had the jet yak moving all over inside salt pond, then it went through the channel, and then it sort of came out here and made measurements outside. And this is the dashboard we got. And sure enough, what do we see? What are all these big, big cells? These are the only Alexandrium we see are these big planozygotes. These are all heterocapsa. This is a different dinoflagellate, a different organism here entirely. But the Alexandrium that are being lost from the pond are these sexual stages, and which means that the cysts then are making it out into the marsh and potentially further. Still, most of them, many of them, are being retained, but we have a dispersal mechanism that we didn't realize. So the cells swim and migrate 2 to 3 times faster in situ than predicted. Cells also vary their migration behavior in these different life cycle stages with very important implications with respect to species dispersal. See, this is a salt pond. You think, oh, that's only applicable to that little system, doesn't matter in coastal waters. Well, I was talking to someone about a bloom we saw in 2009 where we had a big red water bloom off of Portsmouth. In fact, it was Ruth. And in red water, coastal bloom, and we looked at it full of planozygotes. And all of a sudden it's starting to click. Maybe in the rare times we see Alexandrium make real red tides, it's the end of the blooms where these planozygotes are actually reaching the surface and being visible. And sure enough, when we looked at those samples, full of planozygotes. And clearly that red patch was a forming cyst and they were going to drop out. So even in coastal waters this can be happening, and when there's downwelling or movement of the surface waters, that population is going to get moved to different places. So there are, both in the estuaries and in coastal waters, significant implications with respect to the species dispersal. So the final little bit of data from these in situ sensors is when we did a co-deployment of the IFCB shown here. You see this little guy here, that's the IFCB, and the ESP, which is here. And this is Mike Brosnahan, who's been hugely important in all of this work. So this is our ugly platform. We now have 2 instruments on it, and the data are overwhelming that you can get from this kind of a system. And I can't possibly go through all of it. This shows you, first of all, sort of a bloom that could, like all of our others, that seems like we're all set to pull everything out of the water, and then it starts growing again, and then we have sort of a second bloom that happened. Also, all of Alexandrium. You can't really see this, but this is depth and temperature. And so you can see cold water, you can see it getting warmer, and you can also see where the cells, it's hard to see this up and down. Notice that's the profiling winch, and you can see that there are times when the surface waters are warm, there's a real cold period here. So you get a lot of details on the environment that these cells were following. This is that fluorescent trace. And remember that we are following that trace with the IFCB. So we know the temperatures at which these cells were here. We know the temperatures here, and as well as here. So there's a huge amount of information there. Here is that same biovolume plot that I showed you several times before. The cells are getting bigger and smaller. You see this sort of phased division here. You see at the time again then where the bloom is peaking, the cells get smaller, they start getting larger, and then of course we have this second bloom. I'm not going to focus on that one now, but it's the very same story. Large number of cells making these gametes. But the different thing that's in this image, other than a lot of the temperature data here, is shown down here, where we've got the toxin per cell in red shown here throughout the course of that bloom. Remember I said that the ESP can measure toxin as well as cell abundance. Here in this little thin shaded line, that's the toxin per cell for the isolates that we grow from that salt pond, the cultures we have. So we tend to think that they're in this range of toxicity. And so what is our observation? That during this bloom the cells are essentially always more toxic than we thought they should be. They're actually as much as 4 to 5 times, and I'm being added conservative, some of the people looking at the data here are saying 10 times more toxic than we were predicting or than we would think. And this is the in situ growth rate, and you can also see that it's quite high throughout that interval. So basically, Alexandrium toxin content, the toxin per cell, was basically more than 4 first of all, it varied more than fourfold during the course of the bloom. So the cells, we know that it's not like they produce a certain amount of toxin and that's it. We now know that some cells are 4 times more toxic than other cells at a different stage of the bloom in situ. And it was higher for the vast majority of the bloom interval compared to levels observed in laboratory cultures. And similarly, the growth rate, this in situ growth rate, varied 4-fold over just 8 days. This was, I won't try to go back in detail, but we had a real cold snap in here. It got really cold, and we saw that reflected immediately in the in situ growth rate. So it's just sort of like you feel like you're a peeping Tom. You've got a window into the life of this organism. It gets cold and they stop growing. They start slowing down. You get to see their sex life. You do all of these things going on, and this instrument is watching it for us. And it's the variability that we're seeing, and again, think about that. Their growth rate varied 4-fold over an 8-day interval. So all of our models, everything else, assume we can get a constant growth rate at a certain temperature. It's not how it works out there, not at all. So I've mentioned that we've done underway sampling. We've put the IFCB on vessels like this. I've mentioned the jet yak. It's now soon to be put on a wave glider. I don't know whether you've seen those. They're these very expensive platforms that move with the waves, and they can actually move through the water just with the energy that's in the waves, and there's now an IFCB being put on one of those. I've mentioned that the ESP is now being made smaller and put in an AUV so that it can travel and be mobile. And so to wrap all this up, what have I tried to tell you? In situ deployments of ESPs, first of all, reveal these high frequency fluctuations in toxicity that simply aren't captured by weekly toxin monitoring programs. In situ deployments of the IFCBs reveal that the bloom cells swim faster, migrate differently, divide faster, form more gametes, and are more toxic than suggested from past laboratory studies. Basically everything we thought about this organism, we were wrong. Okay, we didn't have good estimates. So deployment of in situ sensors can characterize vital rates and environmental responses far more accurately than laboratory cultures. Now many of you would say, oh, of course that's true, but there's 2 things I'd say to that. The first is, we've been culturing Alexandrium for decades and decades, and we really thought and did think that we were doing well, that we could form cysts in culture, that we can get them to go through a lot of different stages, we could get them parasitized, we can do all of these things, but it's just not the same as what's going on in the field. So now I've told you IFCB and ESP sensors are being modified and deployed on these mobile platforms, enabling underway and adaptive mapping of HAB cells. And so the bottom line is that our ability to understand and forecast HABs, and of course many other marine phenomena, requires that we characterize critical rates and behavioral patterns in natural populations. But what I'm really saying, these need to be generated whenever possible through in situ observations. So all the models, we have all of these things. The framework is there, but we now have to start changing the rates that we're using in them. And that's, some of us have been talking about a big proposal that a number of people submitted to the Centers for Oceans and Human Health program, and one of the centerpieces of our proposal is to get new rates based on these sensors, to go into different environments, not just salt pond, to bring these instruments to other places, start measuring these rates and seeing how common these observations are. And of course I believe that optical and molecular technologies, which are the basis of these 2 instruments, will play a major role in these future developments. But the final thing I just wanted to say is, I'm going to anticipate somebody's question. Why? Why are they growing faster? What is it that makes that difference? And the answer obviously is I don't know. But I can give you some thoughts. The first thing that skeptics and others would say, oh, well, it's bottle effects. You've taken this organism that normally would be out in the environment swimming around, you put it in a container that's got walls, and it's just not going to be the same. Well, I don't think that's the whole story. My current thinking is, and it's best explained with an analogy. We've all heard about if you're trying to get a horse or a donkey to move, you have a carrot and a stick. You can dangle a carrot in front of it, or you can hit it to make it go, one way or the other. Well, in the cultures, we've got the carrots. We've given them all the nutrients we want, nice light, nothing really bad in there, but we don't have the stick. And what's the stick? The stick would be grazers. The stick would be parasites. The stick would be competing organisms releasing chemicals in the chemical warfare that we know is going on out there. And in that environment, I think these cells just behave differently. We think they're just pumped up a little more. They're doing everything a little faster. It's like it's a nastier world out there than in our cultures. So that's my hypothesis, is what we would love to prove someday. And it's certainly the result, the topic of a future seminar, hopefully. So with that, I'm going to acknowledge all of the people and the agencies that have helped to support this work, and I want to thank you for your attention again. Thank you.